All right. Okay, so basically, my talk follows Balázs's introduction perfectly. I also start from this scheme where we start with a sensitive cell, here labeled with the red circle, that becomes somehow resistant over the course of the treatment. There are many different reasons why cancer cells can become resistant, and these are just the cellular factors. Basically, anything that happens to the drug prior to its hitting the target can lead to resistance. And as Balázs has introduced, one major preemptive mechanism is already at the level of the membrane, where these transporters are overexpressed and they kick drugs out of the cells. Thus, these drugs cannot reach the critical concentrations needed for the cell-killing activity. We know that these transporters belong to the ABC transporters superfamily, and as Balázs has introduced, there are three prominent members of this superfamily: P glycoprotein or ABCB1, ABCG2, and ABCC1 that have been shown to be expressed on cancer cells. So what happens is that perhaps when the therapy is started, the majority of the cancer cells do not express these resistance factors, and when at the time of when resistance is experienced at the patient, with the patients, then perhaps these tumor cells are equipped with transporters. But of course, in real life, the situation is much more complex. And if we look at all the possible mechanisms by which a patient can become refractory to treatment, we'll notice that only part of these can be attributed to cellular mechanisms. The cancer cell niche, the microenvironment is another very important factor. The plasticity of the whole system is a very important factor. And you have all these mechanisms which also play a role. So how can we really study resistance by acknowledging also this complexity? A number of years ago, genetically engineered mouse models have been introduced into the field by Piet Borst's group at the Netherlands Cancer Institute. Basically, these animals develop spontaneous tumors, in that case, triple negative breast cancer tumors, because of a induced deletion in epithelial cells. It's a BRCA1 and p53 loss selectively in epithelial cells, and as a result, mice develop mammary cancer or in about 1 to 200 days. What is really important is that these cancers resemble closely to the human triple negative hereditary breast cancers. And also from our perspective, it's important that they can be treated by chemotherapy and they respond to treatment. And after some time, as a result of repeated treatments, they become resistant. So initial sensitivity is also shown on this tumor volume curve. On the x-axis, you see days. Each arrow represents a treatment, and the y-axis shows the tumor volume. And as you can see, treatment has some intermediate effect. The tumors can be pushed back, but eventually the tumor becomes resistant. What is important, and this was shown already in the Netherlands Cancer Institute, that the mechanism by which tumors become resistant against, for example, doxorubicin, is again the overexpression of transporters. So these resistant tumors use the same mechanism as was seen so many times in vitro when we, for example, selected cells with doxorubicins in vitro. So this in vivo model, which on one hand is a very good model for triple negative breast cancer, can be now used to study the evolution of resistance, which is again mediated by PGP. And in this lower figure just shows that PGP activity can be monitored in vivo if you use a tracer, which is a substrate of the transporters. You can see that in tumors that express high PGP levels or ABCB1 levels, the accumulation of this tracer will be reduced as a result of the efflux function. So again, we have a model which recapitulates the evolution of therapy resistance. The mechanism of PG is mediated by PGP, and all these factors, which I in a few slides before I indicated to be relevant in vivo, are also at play here, because this is an in vivo treatment protocol. So you can ask the following questions. Can you now design a therapeutic approach to overcome this resistance? Can you understand the evolution of resistance? And if you do, can you prevent it? And in this talk, I will show you that with this model, you can actually ask now new kinds of questions, which not only relate to resistant clones, but also addresses intermediate stages. So recently, the field is getting familiar with the theme of persister cells. And to introduce persistence, I think I have to go back to one of Balázs's slides, where he showed that if you start with a sensitive tumor cell population and you end up with a resistant population here labeled with blue, there are multiple scenarios or models that you can imagine. You can imagine that initially there was some sort of a heterogeneity, and already at the first during the first treatment you had some cells which were intrinsically resistant, and what you do with the therapy, you just select these out. You can imagine that during treatment somehow a mechanism is induced as a mutation, and then those cells which become resistant then have a survival advantage, and this will lead to resistant tumors. But there is a third possibility, that you start with a sensitive population, and as a result of a treatment, you have a transitory phenotype, which, however, is lost as the tumor cells start to proliferate again. And this transient resistance or persistence is called drug-tolerant persister cells. And much attention is focused today, nowadays, to understand the mechanism by which tumor cells can endure treatment even without a stable resistance phenotype. And this is, if you think about this, this is really reminiscent of the cancer stem cell idea. So these are cells which themselves are perhaps not tumorigenic, but they provide a source for cancer cells, and thus could be considered the root of the cancer population and that Balázs was referring to. Okay, so again, this slide just summarizes some of the information that has been revealed recently about these drug-tolerant persister cells or DTP cells. First, we know that they survive treatment per definition. Second, importantly, they demonstrate a transient phenotype. So when they start to proliferate, this phenotype is lost. And they can be in a quiescent or dormant state for a very long time, or a slowly cycling state for a very long time. And this is better shown here with an other curve that we have in the lab. Again, you have the same mouse model. You have different kinds of treatment. This is a combination treatment, and you can see that after the first treatment, there is a very strong response that endures for more than a month. This is a minimal residual disease stage. You don't measure any tumors, but actually the tumor relapses and is sensitive for a second round of the treatment. So these cells that survive here, again labeled with blue, are the persister cells that we want to study. This recent review that appeared last year summarizes many more characteristics of these cells. So what we wanted to do in the lab is to understand a bit more about these cells. So we established an in vitro assay where we tried to mimic this situation. So we take cells, here MCF7 cells in a flask, and we treat them with very high concentrations of drugs, here doxorubicin, for a few days. As a result, most of the cells die, but some survive. If we look at the flask very carefully, we can see single cells that show a very weird, bizarre phenotype that survive. And after sometimes 1 or 2 months, these single cells start to proliferate again, and we have again a new cell line that we call repopulated cells. So we wanted to study these persister cells to understand, their secret. What we this first slide just shows you the experiment again. So this is treatment, persister stage, and relapse or repopulation. What's important is that when we compare the sensitivity of the initial cell line with the repopulated relapse cell line, the sensitivities are the same. We have here 3 drugs, 3 cell lines. So it's not that the relapsing or repopulating cells become resistant. Again, this is a transient phenotype which enables survival in these cells. The repopulating cells are not resistant. This shows you the time it takes for these cells to recover. You can see that if we take, for example, MCF7, we have 3 different kinds of treatments: doxorubicin, olaparib, or cisplatin, and it takes more than a month for these cells to wake up. So it's a very lengthy process during which time the cells are in a dormant state. Okay, so we collected these cells and wanted to characterize their phenotype. And this is a summary slide here. You can see that we could show that they undergo EMT, they show signs of senescence, they show a number of stem cell markers, a number of epigenetic changes. But for this talk, I would like to focus on the expression of ABCB1 or P-glycoprotein, this transporter. We could show that whereas at the start, the cells did not express any of the transporters, here at very early stages, before the drug was removed, day 6, day 7, a small fraction, subpopulation of the cells expressed higher levels of ABCB1 or P-glycoprotein. This shows you the percentage. So when we treated, again, MCF7 cells shown here with doxorubicin, we could show that at this early stage, about 5% of the cells started to express these transporters, whereas in the repopulated cells, again, transporters were not expressed at all. Similarly, when cells were treated with olaparib and other substrate of these transporters, we could see a similar induced expression in a subset of cells. This induction was not detected when cells were treated with cisplatin, which is not recognized by the transporter. So the big question is, you see no expression, induced expression at the persister stage, and then no expression at the relapse. So can you do something about it? Is this relevant? Can you exploit the fact that persister cells show a transient expression, an induced expression of PGP? So to address this question, you can deploy an inhibitor of the transporter. And this is a complex experiment that we performed in vitro. We had a condition where we just treated the cells with doxorubicin that I showed you before. A second scenario where we added tariquidar, the inhibitor of the transporters, when the cells were treated with doxorubicin. And 2 other scenarios where we, in the first case, we prolonged the treatment of inhibitors in the persister stage, or we only apply the inhibitor after doxorubicin has been removed. And we wanted to compare the relapse time, in vitro relapse time of these conditions. So this sort of Kaplan-Meier curve shows you the relapse time. And I was explaining you that when we apply doxorubicin, the cells, really a lot of cells die, but after about 40, 60 days, in each flask, you see the relapse of the new cell line, the repopulated cells. So the first flask started to repopulate at day 55, but then until this time, all the 4 flasks that we had in the study were completely repopulated. So when we added tariquidar together with doxorubicin, there was no difference in this repopulating time, indicating that there was no effect of just adding an inhibitor to cells which do not express any transporters. But when we added inhibitor to the cells after the drug has been removed, so at the persister stage, you can see that it took much more time for the cells to recover, indicating that induced expression of PGP in persister cells provides a vulnerability that you can exploit by the inhibition of PGP. So this is what this experiment shows you. I understand I don't have a lot of time. Balázs, please indicate if I'm really running out. I just want to show you that we repeated this experiment in our mouse model also. Again, we have a treatment protocol where we just add doxorubicin to the mice. And if you do that, you can have transient success, but eventually the tumor becomes resistant to treatment. Then we combined doxorubicin with tariquidar again, the inhibitor. This is condition 2. Condition 3, where we this is condition when we combined. This condition is when we just add the inhibitor alone without doxorubicin, and then we again in this 4th condition we continued the treatment only with the inhibitor without the drug, again to somehow target persister cells. And what you can see here, this is again a complex figure. You can see that when you don't treat mice, this is the black curve, they succumb to, you have to sacrifice them pretty soon because of the tumor growth. The inhibitor alone, the green line, does not make any difference. It's not toxic. Doxorubicin alone, as I showed you, it results in an overall survival of about 60 days because the tumors become resistant. However, if you maintain treatment with just the inhibitor, this condition here, so when you add doxorubicin and then during the persister phase, the inhibitor, you can really prolong the survival of mice, again showing that persister cells that survive treatment without themselves becoming resistant can be targeted by the inhibition of these transporters. So this brings to my summary, which says that therapy resistance is, of course, a major impediment to successful treatment. Of course, resistance is a complex phenotype based on very complicated mechanisms, of which PGP-mediated resistance might be only a subset. And even though only a fraction of cancers express PGP when they are resistant, we could demonstrate that the success of treatment in vitro and also in vivo with the PGP substrate doxorubicin and can be significantly improved if we target DTP cells with PGP inhibitors. The final slide is the acknowledgement slide. So this work was performed in Budapest and in Vienna. And these are the co-workers and some of the funding. And thank you for your attention. I understand that I was really fast. I was trying to tell you a lot in a short time. So if you have any questions that I can answer, I'm here for you. Thank you.